Hey guys, anybody can hear me? Let me see. Should computer sound? And I'm going to share this. Okay, so let's go through the PowerPoint again. And again, when we're going through these, let me get back here to where we ended. Some of this can look very, very complex, right? So when we're looking at this and you see all these different numbers here, it can look very complex. Just keep in mind that when we're doing it, right, there's always a number on top here that kind of tells us what is happening. So follow those numbers, they go in sequence. The easiest way to memorize and to understand even the hardest concept is to look at the sequence of how things happen. So when we're talking about this, there's a sequence of events, right? So number one, you have an action potential going down our motor neuron, causing calcium to be entering your motor neuron. That calcium causes acetylcholine to be released. Acetylcholine then binds to its receptor. The receptor opens up, and the receptor is a sodium channel. So all acetylcholine receptors are sodium channels. When there's no acetylcholine, the receptor is shut. When there is acetylcholine, the receptor opens and sodium rushes in. So that does happen, and it does allow us at that point to, right, to kind of understand that there is a sequence, right, when you're going through this, and you can kind of read it and kind of follow the numbers all the way through. Now, the reason why I'm mentioning this is because in a couple of seconds, like right here, with this slide number 37, there's a lot of information here. And then the next one, right? The next three, they have a lot of information. So the good news is that that information is very important. It helps you understand what we've been mentioning. So let's go through these right now. And then as we go through it, you can hopefully understand what's happening right here. So let's go through number one, right? Number one, we have an action potential that's triggered. So an action potential that was produced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. And all that means, right? All that means in terms of an action potential right here, right, is that it starts at the neuromuscular junction, and now the action potential is going to activate one segment to the next to the next. So it goes right down the whole plasma membrane with one segment activating the next one. That's why you see this kind of jumping right here, right? So as one segment activates the next one, right, what happens then? It hits a transverse tubule. Remember, this is where transverse tubules become very important. The transverse tubules are parts of the plasma membrane that gets pushed inside. It's part of what we call a triad, right? A combination of one transverse tubule and the sarcoplasmic reticulum on each side. So what happens, right? And this is kind of the answer for the question I had just given you guys, right? Now, I'd still want you to think about it. You have an action potential that goes from one segment to the next, then it has to go down this transverse tubule. As it goes down this transverse tubule, now an action potential travels down the transverse tubule. There's a change in charge right here because there's a nerve impulse there. That nerve impulse going down the transverse tubule causes calcium release. As calcium's released, you only activate the sarcomeres here, right? So calcium's released from the sarcoplasmic reticulum. The calcium that was stored at the sarcoplasmic reticulum is now released. The calcium binds to troponin. Troponin then shifts and moves tropomyosin out of the way. As tropomyosin's moved out of the way, the binding sites open up. As they open up, myosin and actin can interact, and myosin pulls actin to the middle. Myosin pulls actin to the middle over here. Right? So again, as you review this, right, I want you guys to use your own words, but this is the reason why certain Z discs move while others don't. So let's take a look at the next slide, right? And it kind of shows us that once we have opened up the binding site, myosin's going to reach up and pull, right? So you'll notice that the myosin, there's no more ATP already. Why? It's because before it has a chance, all right, it's already cocked its head before the myosin and actin even interacts. So as soon as you have one cycle of contraction, the myosin head receives a new ATP. It's going to cock the head right here for the next cycle. If there's still open binding sites, what's going to happen then is myosin is going to reach up and pull again. All right, so with each pulling right here, with each cycle, right, so this guy's going to pull one time. Well, the ATP is going to drive that movement. Now, the weird thing is this. ATP allows us to contract. ATP also is what allows us to relax, right? Now, before we talked about it, all right, if you guys can understand this, right, again, very complex. It seems kind of overwhelming, but follow the numbers, right? And it kind of explains what happens. Number one, motor neuron and action potential as my brain tells my muscle to move, right? What happens then, right? You have an action potential going down the motor neuron as it goes down into the terminal, the synaptic bulb area right here, right? That action potential causes calcium to rush in. Calcium rushing in causes acetylcholine release. Acetylcholine binds to its receptor, which is a sodium channel. Sodium rushes in, allowing us to hit threshold. Now, again, they got lazy here and don't show voltage-gated sodium channels. Keep in mind, voltage-gated sodium channels, they are the ones that cause action potentials, right? Not the acetylcholine receptors. Acetylcholine receptors allows us to hit threshold. But once we hit threshold, it's a voltage-gated sodium channels that allows us to have this action potential that moves from segment to segment. As it moves down the sarcolemma, segment to segment, it's then going to hit a T-tubule. As it hits the T-tubule, the action potential goes down the T-tubule, which causes the calcium release from that segment only. Right? As it goes up the T-tubule, it causes the next sarcomere to undergo a calcium release from the sarcoplasmic reticulum. Right? So there's a sequence. Right? In other words, when you have a nerve impulse in a muscle, you don't just stimulate the whole muscle all at once. That doesn't happen. Instead, right, each segment causes the next segment, causing the next, causing the next. Now, what happens is, instead of everything electrically depolarizing at once, you have certain areas depolarizing, then the next area depolarizes, then the next one. So there's a sequence of it right here. So then you have a sequence of calcium release, and this one will release calcium first. So this sarcomere here will depolarize and it will be able to mechanically contract, while the other ones here are relaxed, right? Again, as you go through this, it kind of should help you figure out the answer that I posed before. Why is it that some Z discs move while others don't? And it has to do with the sequence of these action potentials. Right? See if you can write it out. And I'll write the actual like question on our discussion board. Okay. Again, you know, it'll be anonymous to everybody else when you're writing it on the discussion board, but I will see if you guys got it right or wrong. Right now, we talked about how muscle contracts. Muscle contracts by calcium release, and calcium is the link between what happens electrically and what happens mechanically, right? Again, electrically is an action potential. An action potential going down the T tubules causes calcium release. Then calcium causes the tropomyosin to move out of the way, which then allows you to have the mechanical contraction. So there's that kind of, you know, two parts, right? Electrical and mechanical. Calcium links both of them. So that's why calcium is so important. Now, what else do we see? For muscles to relax, you need brand new ATP to bind to the myosin head. Once it does, ATP allows the myosin, fresh ATP, allows myosin and actin to dislocate from each other. As it does so, then we can relax or we can contract again, depending on if calcium's available. Now, there's a couple proteins I want you to know. This is one of them. The protein calsequestrin, found in the sarcoplasmic reticulum, binds to calcium. It allows us to remove it from the sarcoplasm. Calsequestrin is found in the sarcoplasmic reticulum. Sequestering is moving something out, right? You guys have heard of, probably, if you watch any TV shows, right, any kind of crime shows, they talk about sequestering a jury. Sequestering means you remove that person from their home and you put them into a hotel. So then you can kind of control what they're watching. So sequestering is removal. Calsequestrin is calcium removal. And what it does is a protein that binds to all the calcium, so then you have very little of it in the cytoplasm or the sarcoplasm. So at rest, all the calcium are bound to this protein found in the sarcoplasmic reticulum. When you want a muscle contraction to occur, all the calcium on the calsequestrin is released, right? Then it leaves the sarcoplasmic reticulum and enters your cytoplasm. Then it binds to troponin. Troponin moves tropomyosin out of the way. Again, I want you to be writing, if you can, right? Have a sheet of paper and write out the sequence as you kind of just think about it. See if you can write out, right? Again, always start electrically and then mechanically. What causes the electrical activation? What happens as a result? Right? The whole point of an action potential is to just release calcium. That's it, right? The calcium then causes the mechanical number 2 step, right? So keep that in mind as you go through it. If you can kind of run it through in your head, all these kind of steps, you'll be in pretty good shape. So besides ATP, and it's weird, ATP provides the actual fuel for muscle contraction. It also allows you to relax. And we'll talk about what happens in a second when we don't have ATP. So here for relaxation, number 1, we need to get rid of acetylcholine by acetylcholine esterase, right? That stops further action potentials. Then we need to get rid of calcium and move it out by binding it with calsequestrin in the sarcoplasmic reticulum. Then the last thing we need to do is fresh ATP must bind to the myosin heads. Once it does, myosin can pull away from actin. They can dislocate from each other. Once they dislocate, and now there's no more calcium, because the calcium right here has been removed, well, now tropomyosin moves back in the way. Tropomyosin moves back in the way, myosin can't reach up and make contact again, you can relax. Perfect. Now, in terms of these twitches, all we've done so far is cause a muscle twitch. In other words, I have an action potential that causes calcium release, that causes a muscle contraction. All of that only causes a little twitch, and that's it. Not a very strong contraction at all, right? So it causes that little eye twitch, that's all. Now, how do we get a muscle to contract so then we can move, so then we can walk, right? That requires more than one action potential. And right, that's what we're going to talk about next. How do we get a muscle to contract stronger, more smooth, right? And it has to do with a couple things. Number one, it has to do with motor units, right? A motor unit is a single motor neuron and all of the skeletal muscles it goes to. Innervation means control. So when you see the word innervation here, right? So a single motor neuron and all the muscle fibers innervated. So it's one motor neuron and all of the skeletal muscles that it controls. So if you look at this picture, here's one motor neuron, and it doesn't just go to one skeletal muscle, right? It'll go to one skeletal muscle over here, and then another one over here, and then another one over here, and then another one over here. So one motor neuron can go to more than one skeletal muscle. So one motor neuron can control more than one skeletal muscle fiber. Now, a motor unit is the motor neuron and all of the skeletal muscle fibers it goes to. In large muscles, one motor neuron activates lots of muscles, thousands of different muscle fibers. Why is that important? You activate one motor neuron and 1,000 muscle fibers will contract. That gives you a lot of strength, right? So it makes sense, like your quadriceps muscle, very big and strong. Well, you want strength over precision. So one motor neuron contracts a lot of skeletal muscle fibers. So when that motor neuron activates, thousands of muscle fibers contract, pretty strong. Now, for your quadriceps, you can have lots of motor units, right? Lots of motor units with lots of muscle fibers, right, for each motor unit. So then what we're gonna see is when I want contraction, one motor unit, one motor neuron fires, all the muscles contract, and then other muscles of that quadriceps are in a relax mode. So then, right, once the one that's been activated contracts for a set amount of time, well, then it has to relax or else it's gonna fatigue. So other motor neurons fire, activating other muscle fibers the next time. So then you can turn certain parts on and off, and it allows us to run like in a marathon, right? Now, for large muscles, one motor unit goes to lots of muscle fibers, thousands of them. For small fibers, like in your thumb and your fingers, right, in your hands, well, those muscles don't need to be super strong. They need to be more precise. Think about what happens if you're typing or playing the piano. If you're playing the piano, those different keys, right? You guys have probably seen pianists kind of move their fingers, and it's almost artistic in how they're hitting the keys. Well, what happens is you need more dexterity, more precision, right, for the movement of your fingers. So we're not quite as worried about strength, but we're more focused on precision. So for the small muscles, one motor unit is one motor neuron would go to very few muscle fibers. So then you can kind of control the exact space, right? The exact location of your fingers as it moves from one point to another. You can imagine if you are doing surgery, right? You need to have incredible precision so then you don't cut something accidentally. So you don't have to worry as much about strength. You just want more precision in the fingers. Next, right, basically telling us what happens if there is, right, if you have a subthreshold, threshold, or suprathreshold stimulus. Meaning what? What happens if I have an action potential or a motor neuron and I don't hit the actual, the threshold, minus 55 millivolts. So we say it's subthreshold stimulus. Well, if you don't hit threshold, you don't hit minus 55 millivolts, nothing happens. There's no action potential, there's no twitch, there's no contraction, nothing. You have to at least hit threshold. If you have a stimulus that's 30 millivolts, now remember, we start off at minus 85. So a 30 millivolt stimuli only gets us to minus 55, right? At minus 55, you have a twitch then. Just one twitch and one action potential. That's it. Now, how can I have one action potential that's different from another one? Meaning what? If I'm lifting weights, right, and I'm just warming up and I'm doing 5-pound dumbbell curls, right? How do I get my muscle to contract differently between 5-pound muscle curls, and then when I'm starting to work out for real, I have 50-pound muscle curls, right? So how can my brain tell my muscles to contract slightly different then? Well, it's because of this, right? This supra-threshold stimulus. And what we're going to see is when we want more muscles and more strength of contraction, we don't have a stronger action potential. There's no such thing. So if I'm lifting 5 pounds versus 50 pounds, right? I have the same level of an action potential, right? That's it. An action potential is just an action potential. It goes to positive 20, come back down. But instead of having a stronger action potential, I will have more action potentials. So the stronger the stimulus I need, the stronger I'm lifting, the more weight I'm lifting, right? I'm gonna have more action potentials to cope. So then I have more strength. So that's a big difference here. So in order to lift that 50-pound dumbbells, I need to activate more motor units. The more motor units, the more muscles that activate. Kind of makes sense, right? The more muscles, the stronger the contraction. So not only do I need more motor units when I am lifting heavier weight, but I also need to have more action potentials. So then I can stimulate the muscles faster, right? And what we see is when we have an action potential and only one action potential, I have a very weak contraction. But when I have lots of action potentials in a short time frame, each action potential after that initial couple will trigger a stronger stimulus. See that? So the first one, tension's pretty weak. The second one, action potential, right? I can increase the tension or strength of that contraction. The third one, then I hit maximal contraction. So if I want to lift something really heavy, I need two things to happen. One, I need to activate more muscles. So I recruit more motor units. In other words, more motor neurons fire that causes more muscles to activate. More muscles, more mechanical strength. The second thing is I also need more action potentials, right, occurring in that motor unit as well, all right? And what we're gonna see is the more action potentials, and this is what we call motor unit and wave summation over here, right? So this best example of it, I have one action potential. What happens? Just a twitch contraction, pretty weak. So that's not good enough for lifting 50-pound weights. So how am I gonna lift 50-pound weights? Number one, I'm gonna need more muscles activated. So I recruit more motor neurons. And as I recruit more motor neurons, I recruit more muscles to be involved. Second thing, that's number one. The second thing is, right, to have it nice, strong, smooth contraction, I also need more action potentials. So here is one action potential. I have a twitch. Before I have a chance to fully recover, I have a second action potential, right? The second action potential causes that contraction to be stronger. See that? Even then, it's still not nice and smooth, right? If we depend on this, we'll have like the herky-jerky movement of a muscle. Not good. We want nice and smooth. So how do we get nice and smooth? Lots of action potentials in a very short time frame. Here, we have lots of action potentials, but it's too long, too far apart. So I have an action potential here, I have a muscle contraction. and then I let it relax a little bit, and now I have a second action potential. Yeah, the muscle contraction is stronger, but there's a little bit of a lag, right? So the movement isn't smooth, it's more herky-jerky then, right? So if I want nice, smooth, strong movement, think about what happens when you're lifting weights and you can handle the weight. It's nice and smooth, that movement. In order to get that, I have lots of action potentials in a very short time frame. So here's time right here. Take a look at how short the interval is. I have an action potential here, and then it moves up, and before I have a chance to relax at all, I have a second action potential. So then the second action potential causes a building of strength. Before I relax, I have a third action potential. So I keep building because I have so many action potentials, right? So the faster the action potential comes, and the more action potentials, the more buildup of strength we have. We call this a tetanic contraction. So tetanic contractions are good. That's actually a good thing. That allows us to have smooth, strong contractions. This is not tetanus, like when you get the disease, right? That's bad. Here we're talking about nice, smooth contractions. That is the target when we're lifting weights or when you're even walking, because walking is lifting your body weight. You can imagine if people are walking and they're twitchy, right? You have a chance to fall if that's a case. So we want nice, smooth, strong contractions. And that's what this is with fused tetanus, right? Again, wave summation is nothing more than the buildup of tension, the buildup of strength with each action potential. That's the name of wave summation. The best example of wave summation is fused tetanus or complete tetanus, meaning that when you have an action potential, the first one, you have a building up of strength. Before you have a chance to relax, we have another one. You build up more strength. Before you have a chance to relax, you have a third one, and then a fourth one, and then a fifth one. So then there's never this herky-jerky movement. There's just nice, smooth movement until you have maximum contraction strength. Perfect. Now I know, all right. Now, in terms of muscle length and tension, right here, keep in mind that if you've ever seen an athlete, I remember when my son was a little younger and LeBron James was playing in Miami and he came up here for a game and we had tickets and we went to watch him play. And what's crazy is like we were there early and all he did was stretch. That's all he did. We thought he was going to do some dunks or something. No, all he did was stretch. Why am I mentioning this? Stretching is going to be important because of what it does to our length versus tension. In other words, there's an optimal length of your muscle. So then when you contract it the next time, you'll have a very good, strong contraction. So let's take a look at this. And pictures are usually pretty good to show it. Right here, right? When you have an optimal length of the muscle, that means that all of the myosin can interact with actin. See that? There's every single myosin head is covered by actin. Then, right, what happens is, right, there's also lots of space for the actin to move. So you need both, right? You need lots of myosin heads that can interact with actin. That interaction between myosin and actin is called a cross bridge. Lots of cross bridges and lots of space, right here. So then you can kind of move everything to the middle. When we contract and our muscles are at the optimal length, we have a very strong contraction. You can see as number 2 over here. Very strong contraction. Now, what happens when you get tired? Let's say you are working out and you're doing bench press. And I see this at the gym, right? And you see kids bench pressing, and then the next day they bench press again. So what happens is when you are, let's say, doing push-ups or bench pressing, or when you first start, your muscles are optimal length. So you're able to do it pretty easy. You can do a lot of push-ups right away. And then after a couple sets, right? You did 50 push-ups. Now you're going to relax a little bit. Now you're going to do another 50. What happens is after you've done a couple sets of 50, right, you get to this area where the muscle starts to shorten. Because I've heard of videos where they talk about muscles getting a pump, right? This is a muscle getting a pump. It's pumped up because you've contracted so much that the muscles are naturally shorter because you've already contracted it so often because of the exercise. Well, the good news is the muscle looks huge, right? The pump. The bad news is if you're going to try to do another set, take a look. Is there any space here anymore? There's no more space between the actin. So then if I have a muscle contraction and the muscle is already this short, myosin can pull on each side, but there's no room for the actin to move. So the next contraction is very weak as a result of this. We call this a crumpled muscle. By the way, this is why you feel the urge to stretch out. So after you've done a couple sets on the bench press or bicep curls, what happens is you feel that urge to kind of stretch that muscle out. Why? Because after you've done a couple sets, your muscles are very crumpled, very short. By stretching, you're trying to physically move the muscle away from each other. So you're going from number one back to number two a little bit. So as you physically pull the muscle away from each other, the Z disc away, now there's a little bit more space. And now the next time you contract it, your next set can be pretty good, right? But that is why we have that urge to stretch that muscle. Now, most crumpled muscles, eventually, no matter how much you try to stretch it, right, you're not going to get it to the optimal length already. You're tired, and even if you stretch it for a long time, right, the next maybe 1 or 2 reps might look easy, but you will fatigue quickly. The what we see here, number 3, is a muscle that's overly stretched, torn from each other, right? When that happens, there's a lot of space for actin to move to the middle and shorten. But the myosin heads are no longer covered. See that? Because the myosin heads are no longer covered, the next contraction is going to be incredibly weak because you have all these myosin heads, they're not pulling for that muscle contraction. They're not doing anything because they're no longer making contact with actin. So only these little ones right here. Here, it's only one myosin head on each side that's making contact. You can imagine if they contract, only one of the heads pull. Not very strong. That's why you see such a small tension percentage here, right? Again, we want optimal length right here. The optimal length means that we have, right? We have an optimal length. We have lots of room for the actin to move medially, and all the myosin and actins can interact with each other. That's perfect. Right? As we contract the muscle more and more, we start becoming more, right, crumpled, or the muscles become shorter. And while all the myosins can interact with actin, there's no more room for them to pull. So the contraction is slightly weaker. If you overstretch it, there's lots of room for actin to be pulled, but not enough myosin actin cross bridging. So the myosin heads can't reach up and pull to the same strength because you have some of them that aren't even making contact right here. Right now, some of these you might have heard of in terms of muscle contraction. This kind of question is very kind of easy question to ask on a multiple choice. So what is an isometric contraction, isotonic, things like that. Isometric, no change in length, but tension increases. It's like when you are, playing a sport and you are messing around and your coach tells you to just stand against the wall and sit. So all you're doing is just putting your body weight on the wall, right? And you're sitting there with your back towards the wall. What happens? There's tension, but you're not moving. That's an isometric exercise. Planking is an isometric exercise. When you're doing the plank, you're putting your elbows down, right? And you're creating some pressure in the abdomen and your torso. Are you moving? No, right? But there's still tension. In isotonic exercises, there's length change, but tension is constant. Think of a bicep curl. When you think about a bicep curl, all we're doing is putting weight on our hands and curling your arms upward, your forearms up. As you curl your arm upward, if you have 25-pound dumbbells, it doesn't magically change to 30. So the tension stays the same, 25 pounds all the way through, right? So, but the muscle changes as you contract it. There's concentric when you're actually causing the movement, and there's eccentric, which is like the negative rep when you're slowly moving that weight back down. So concentric movement is part of isotonic. So in the bicep curl, that's when you're actually moving that weight up and curling it. When you're doing a push-up, that's when you're pushing up from the floor. That's a concentric movement, right? The eccentric is when you're moving it to its normal position, right? For a bicep curl, that's when your arm goes back down and extends. For the push-up is when you go downwards towards the floor. That's an eccentric movement. All right. In terms of physical contracture, I'll talk in terms of this, right? Muscle fatigue, psychological fatigue. There's muscular fatigue and there's a synaptic. Now, psychological is when you haven't slept all day and now, or all night, and then you go work out, right? You're tired from not sleeping, or you had, a bad day at work, right? And psychologically, you're just not ready for that workout. It can give you more muscle fatigue and your workout might not be as good. Muscular fatigue is when you have already done the workout and now you've completed and you're tired, right? ATP depletion, you can still move the muscle, but it's not with the same strength. The worst is a synaptic one. That means there's no more acetylcholine. Causes issues related to walking and balance. You guys have probably watched videos of people, right? Usually they're like crazy triathletes, right? There's a video of this triathlete, and there was two twin brothers from England. One was about to win this triathlete grand championship. I know, I can't believe that, triathlete grand championship. But what happens was this, they have like a series of races for triathlons. And what happened with this brother is he was winning, all right? This was the last race. All he had to do was finish in the top 10 and he'd be crowned champion and win like $50,000. Well, he was in first place with about 100 meters to go. And you could just see right when he was about to run. In triathlons, it's swimming first, then biking, and then a marathon. What happened was he was 100 meters away. He was in first place, and then you can just see his legs starts to shake, right? That's synaptic kind of fatigue. What happens then, right? His muscles, his legs, right, just faltered. He couldn't even sustain his body weight. He fell down. As he falls down, he got back up, and he fell back down, right? What happened? 11 people passed him up, right? And then by the time he finished, it was too late. He didn't win the championship because of it. Eventually, he couldn't finish. He was trying to crawl, and he still couldn't crawl. His brother had to help him finish and help him out. That's a kind of synaptic muscle fatigue where you can't do anything, even though you have the mental power to kind of want to do it, right? Physically, there's just not enough acetylcholine release. You cannot cause it to that muscle to work at all. So for this guy, he must have fallen like 3-4 times and then crawled and then stopped crawling because he couldn't even do that. So that does happen. And the take home is don't be a crazy triathlete. The last thing we'll talk about now is this, which is a little bit of rigor mortis and fast and slow twitch fibers, right? So rigor mortis. And with rigor mortis is development of rigid muscle several hours after death. Remember, in terms of ATP, ATP is required for muscles to contract and relax. So what happens when somebody passes away? When somebody passes away, for the first hour or so, the muscles and the body should be nice. They should be, it shouldn't be tight, right? You shouldn't go in a state of rigor yet. But after a few hours, what happens is the sarcoplasmic reticulum starts to become a little leaky. As it becomes a little leaky, a little calcium starts to get released. The calcium then binds to troponin and it moves tropomyosin. Remember, skeletal muscles have glycogen stored in the muscle. Skeletal muscles have oxygen already stored as myoglobin. So it has all the fuel. And now you have calcium there. And what happens is after the first couple hours, right, the muscles start to contract, right? As it starts to contract, the body becomes more and more rigid because of that muscle contraction. That's rigor mortis. Eventually what happens is, right, all the calcium starts leaking out because the cell is dead. As all the calcium starts to leak out, the muscles start to contract and becomes more and more tight. Now, eventually, you don't make any more ATP because you're dead, right? Because you're no longer alive, and all the old ATPs that you had stored up, that gets used up after a few hours. So then people go into this state where they're completely contracted and no more new ATP is available. So they're stuck in this kind of, their body is stuck in a phase where it's just rigid and tight. That's rigor mortis. Now, an example of rigor mortis as well is the reason why we age meat, right? So what happens is aging beef, if you've ever seen movies where they have like a side of a cow hanging, well, when we harvest the animal at a slaughterhouse, what's going to happen? If we harvest, we kill the animal, we're going to have rigor mortis of the animal. So the skeletal muscles, if you wait a couple hours, are going to become really tough and thick, very hard to eat. So we hang it for a month or 2 weeks, right? We call it dry aging it, right? What we're really doing when we dry aging meat is waiting for rigor mortis to go away. Eventually, after a couple weeks, the proteins, actin and myosin, start to dissolve, and then the meat isn't as tough. I know, it's kind of gross, right? So when you go to Sendix and you see meat that's 30-day old, right? The longer you kind of age the meat, the more tender the meat should be because the proteins start to dissolve with that increased time. And we're waiting for rigor to go away by aging it. I know it's kind of gross. All right, now in terms of the, the ATP and the, let me see if I can move this out of the way, energy sources right here. Keep in mind, just very quickly discussing these and what are their function. All right, so there's 3 primary sources of how we can produce ATP. There's creatine phosphate, anaerobic respiration, and aerobic respiration. Obviously, aerobic respiration is the best method, right? In aerobic respiration, we take oxygen and glucose, and through aerobic respiration, which is, and hopefully some of you guys have even heard of this before, which is a combination of glycolysis, then the citric acid cycle or the Krebs cycle, and mitochondrial transport. All those 3 activities allows us to produce a lot of ATP, right? 36 ATP for 1 molecule of glucose. That is the most efficient way of producing energy. Now, the bad thing is it takes time, right? It takes time for your body to use aerobic respiration. Once you use it, you can use it for hours. Think about our triathletes, right? Much more efficient. It allows them to produce a lot of ATP for hours and hours. So then they can exercise for hours and hours. But it takes a while to get your breathing right. Now, creatine phosphate is the fastest alternative. So if you're ready to sprint, right? Sprinting, a 40-yard dash, that uses creatine phosphate. So if you're running a 40-yard dash, you might run it in 6-7 seconds. That's not a very short, that's a very short time frame. You will not have time to have aerobic respiration as your fuel source. Instead, you use something that's readily available, creatine phosphate. So just understand that creatine phosphate allows us to produce ATP for very short bursts of energy. Anaerobic respiration is very intense exercise for a short period of time. You're sprinting, and usually people say like the hardest race is like the 400-meter run, right? In the 400-meter run, it's much longer than the 40-yard dash. So in the 40-yard dash, usually athletes can do it in 4 or 5 seconds, right? Even the best athletes in the 400-meter, they're gonna take 45, 50 seconds. So a lot longer than the energy from creatine phosphate. So instead, they have to use this, right? Anaerobic respiration, right? They're sprinting for as long as they can. And by the time they stop at 45 seconds, there isn't time to use aerobic respiration. Now, most of us have done cardio before, and it's hard when you first start doing cardio because you need to take a lot of breaths. Your respiration normally is 8 to 12 breaths per minute. Once we start exercising, you're on a treadmill, you're on elliptical, your respiratory rate can go up to 30 breaths per minute, right? But it takes a while for you to get it up and get comfortable. If you're sprinting and then you stop right away, you never use oxygen as a fuel source. Instead, you use anaerobic respiration, and you produce a lot of lactic acid as a result, right? That's why you see people on a 400-meter dash, a lot of time they'll pull up because they have cramps, because instead of, all right, they'll use up creatine phosphate in the first 100 meters, and then by the time they get to the end of the race, they haven't transitioned to aerobic respiration yet. It takes about 3 to 5 minutes to transition to that. Well, for them, they're only running 45, 50 seconds. So they're stuck with this one, anaerobic respiration. And it just gives them enough energy to finish off the race. And some people, not even enough energy. And they start producing lactic acid, and they start to cramp up and come up lame. Right? So just kind of review that fuel source for what type of exercise. And the last thing I'll talk about today is the slow and fast twitch fibers, right? And this has to do with meat. I know, meat again, right? So I'm gonna talk about it in us, and then in chicken, and then duck and geese, right? So slow twitch fibers, also known as slow twitch oxidative muscle fibers, are what we call type 1 fibers. With both of these fast and slow twitch fibers, with all of these, there are advantages and disadvantages. Slow twitch fibers contract more slowly. That's a disadvantage. They're smaller in diameter, so they're not going to be usually as strong, but they have better blood supply, more blood vessels, more mitochondria, and a lot of myoglobin. Because they have more mitochondria and myoglobin and more blood vessel, it's also more fatigue resistant. So what we see with slow twitch is that they allow you to contract for long periods of time, right? We find it in humans, our postural muscles. So then you can stand for hours at a time. You can sit for hours at a time, right? Also, we see it as part of the hamstrings and the quadriceps. Now, in chicken, it is a dark meat in chicken. Now, if you've ever eaten a chicken thigh or a drumstick, right? What we notice is that the drumstick and the chicken thigh, it looks dark, right? When you cook it. Why does it look darker? It's because of the myoglobin, right? The more myoglobin, the more redness. Once you cook it, it turns darker in color. So in chicken, right, the dark meat is what allows them to run for a long period of time. But if you look at the chicken, right, the breast tissue has more of the classic fast twitch, right? That is our fast twitch 2B right here. The classic fast twitch fibers responds rapidly to nervous stimulation, so they contract right away, contain myosin that breaks down ATP more rapidly, so they have more cycles, right? The bad news is they have less blood supply, fewer mitochondria or no myoglobin. Unlike slow twitch up here, if you look at a chicken thigh, you'll notice that not only is it darker, but it has a lot of blood vessels. I know, it's kind of gross. But next time you look at it, all the kind of stringy things that we see on a drumstick or thigh, those are blood vessels. When you look at the chicken breast has lots of fast twitch, right? It should have very little blood vessels. So when you see it next time, take a look when you go get some rotisserie chicken, right? Take a look at the chicken breast. See if you see any blood vessels. You shouldn't, right? Now, what happens is because there are no blood vessels and there are no or fewer mitochondria and there's no myoglobin, what happens is they burn out pretty quickly. They can't contract for a long period of time. Now, what happens in a chicken? In a chicken, we know everybody likes eating the chicken breast. So we have modified chicken. So then the breast tissue is almost all fast twitch fibers, right? They don't fly. Because they don't fly, what happens is, right, the muscles themselves there no longer have to contract for a long period of time. Now, when you compare it to duck is basically all dark meat. If you look at the thigh of the duck, it's dark meat, just like in the chicken, right? If you look at the wing and the breast of a duck, also dark meat. There's very little white meat in duck. The reason why is because they have to fly for long periods of time. And because they have to fly for long periods of time, they need more fatigue-resistant muscle structure there in the breast. Now, another other example is turkey, right? Turkeys that we find in farms have a lot of fast twitch muscles in their turkey breasts. So they've been modified, right? So turkeys that we see in the farms, they don't fly anymore. But wild turkeys, they can fly. So if you've ever seen a wild turkeys can fly. They can fly right into a tree. I remember going with my dog, all right? And we were at a park and he chased after a cluster of turkeys and the turkeys all flew away. I'm like, wow, they really can fly, right? So wild turkeys can fly. Why? Because wild turkeys have lots of slow twitch fibers. And that's what natural turkeys have, right? So if you would eat like a wild turkey and eat the wild turkey that is, that is not modified, and you eat the turkey breast, it'll be different than the turkey breast that we see in the farms. The farms, everybody likes a white meat, so they've modified the turkey. They can't fly anymore as a result. Now, keep in mind, this last part is very important right here. In our bodies, all muscle groups will have combinations of fast twitch and slow twitch fibers. So when we say our postural muscles, our erector spinae muscles have more slow twitch fibers, right? It means that they have some fast twitch, but they have more slower twitch fibers found there. When we say our calf muscles, right? Gastrocnemius, soleus. Those muscles usually have more fast twitch. What does that mean? That means it has more of the fast twitch muscles, but even those muscles have some slow twitch fibers as well found there. So there's always different combinations, right? Now I'm just gonna, stop right here. And I know it's about 3 o'clock right now. Do you guys have any questions for me?
Thank you. I don't have any questions.
All right. Again, email me. I will be online as well, and I'll post like a online office like this, like we've been doing, right, to everybody. See if anybody have any questions starting tomorrow morning. Okay, and actually tomorrow afternoon is what I got people saying. So if you don't, I'm gonna post the lectures online, right now, and I'll post the last part, which is smooth muscle and types of muscles, right? Also online. So then you guys can have a chance to review it before you have to take the test. Again, I will release the test this Thursday. You don't have to take it. You can take it anytime, but you will have 70 minutes to complete it. once you start.
Okay.
All right.
Okay.
I'll see you guys
I
next
have
Monday.
one question.
Yes.
For the test, it's on chapter 9, the beginning of chapter
8 and
9.
9. Chapter 8 and chapter 9. Yep.
Okay.
8 is on the joints. It should be pretty, that's the easier one. That's a little bit more factual, you know.
Mm-hmm.
So look at the highlighted material on that, guys. And again, I have videos on that already. So take a look at that on the videos online. Now, I don't think I'll have a lab practical until until the week after we come back from break. So I think would next week, Monday at 3 o'clock be okay with you guys to lecture again next week?
3 o'clock?
At
Yeah,
3
I'm fine.
o'clock?
I am fine.
Right? All right, so I'll send an invite for that. If you guys want more time as well, email me, all right? And I can set up more times that we can do this, okay?
Okay. Thank you.
Okay.
All right.
Thank you,
All right,
Dr.
thank
Lee.
you.
I'll see you next Monday.
All right,
Thank
so
you.
guys, be safe, stay
You
healthy,
too.
okay?
Thank you. You too. Bye-bye.
Bye.